at this event today. Unfortunately, I had a prior commitment rounding at one of our primary care locations. Otherwise, I'd love to be up on T14 with you. This is a pretty incredible moment. The development of an FDA-approved drug, Dupilumab, by Mark Rothenberg and his amazing laboratory team is just something that can only happen at a place like Cincinnati Children's. The combination of a brilliant investigator with persistence and resilience over the number of years, great collaborators and the facilities and the team that came together to make this happen really is something that we as an organization should be incredibly proud of. And Mark, you and your team really are an example of what Cincinnati Children's is all about. We come together to provide great care for kids and to have brilliant researchers like yourself looking at how to make tomorrow better for kids. And when I look at when you started this work 20 years ago, I'm blown away.
Recording in progress.
For my youngest child's high school graduation, and when I think about it, she wasn't even born yet when you started this work. So I just want to thank you and the team for that tremendous persistence. Children and adults with eosinophilic esophagitis are going to have a better tomorrow because of the work that your team has put in over the years. Thank you again and have a great day.
Can you hear me okay? Yeah. Well, thanks a lot for joining today. It is a wonderful occasion, and it's nice to be able to sit back and bring the group together to enjoy and reminisce and celebrate what we've achieved here. But before starting, I do want to just think about something, which is I just want to draw your attention. I don't know if any of you have been to a traditional Jewish wedding, but the reason I bring it up is because at the end of the wedding, despite all the happiness and all the celebrations, the last step to finalize the wedding is the Jewish the man steps and breaks a glass cup, and it's actually the final moment of the wedding, and the ceremony, and everyone claps, and the wedding is consecrated. And the question is, why does that happen? Well, in our tradition, despite great times like today, there's also a dampening, of life, and to understand that the world's not a perfect place, and that our job in the world, even at the highest moment of your life, is to remember to make the world a better place. And I think that this is something we should do today, because especially what happened in Texas this week, to think that we're celebrating here at a children's hospital, while at the same time child health is really in jeopardy, and there's some tragedies, and we need to balance that. And certainly as people who work at a great children's hospital like ours, we need to step up to the plate to understand how a pediatric individual could murder other young kids. And this is, I think, a major health problem that this hospital will be addressing and is addressing. And as you know, there's a strong focus here on mental health, which is certainly very important. So I just wanted to start off by saying that we know that raising children takes a village. And really, to think about it, this is probably part of the problem that happened in Texas, with the man that shot all those kids. Probably the village wasn't intact when he was growing up. But I think our job here at Cincinnati Children's is to improve our village, and I'd say developing a drug, like we're talking about today, takes similarly a village, and that village is Cincinnati Children's Hospital, a great place that comes together to make something like this happen, because it doesn't happen just because of me. That's absolutely true. I happen to be here a long time, and from the beginning, and I help all of you wonderful people bring their ideas to fruition and carry out the projects. But it really takes a village, and it takes a scientific community that we have here that's wonderful and really provides the environment to optimize probabilities of success, and to think about medical problems holistically, to make sure that we're focusing on the right areas, but also optimizing and taking advantage by leveraging the strengths of the hospital. That includes a number of excellent people, which I'm going to thank today. But before doing that, I want to say the most important person to me has been my wife along the journey. Joy, she may be logged in. She's been here every step along the way since we got married when I was an intern, and I haven't stopped working since. She knows me. There hasn't been too many breaks. She's been there every step along the way, and luckily my family, the whole family, has been supportive, including my parents, laid a foundation and helped me in a lot of ways today think about priorities in life, certainly helping other people. I want to start just briefly mentioning where we stand today. Can you see this slide? No, not yet, but this work that we did that contributed to eosinophilic esophagitis and the development of this first drug started in 1999 when I was here for a couple of years at that point and working on another area of research. When Phil Putnam came to see me when I was in my small little lab, and he exposed me to this medical problem, and it wasn't too long before we started meeting with Margaret Collins, who both of them unfortunately aren't here with us in this meeting, but I'm sure they'd be very happy to, and they are happy with the developments that took place. But it was this small group of us, 3 faculty members, that began to form ideas about eosinophilic esophagitis, and of course I was mainly interested in the research and understanding the disease, and they were more involved in the clinical aspects. But over the years, we really started to change the way people thought about this process. Nobody knew a lot about it. There was 1 or 2 papers back in the 90s on this disease, but it was really thought, conventional wisdom, that this was an esophagitis that was caused by acid, like other forms of esophagitis. And it was thought to be a severe variant of acid-induced reflux disease. It was the work that we started to do here back 2 decades ago that showed that EOE was related to allergies, and that we can induce this disease in mice by exposure of mice to allergens in certain manners. That was work that was originally done by Neil Mishra, who also subsequently showed that we can take IL-13 in early experiments and administer it to mice, and those mice who develop an esophagitis. And that was an observation that we began to understand in terms of the connection between the type 2 immune response and the development of EOE, particularly related to respiratory allergies and the connection between the lung and the esophagus. It was around that time when patients who played such an important role in this development in so many ways, but it was particularly Ellen Kodroff who approached me through an email about her daughter in Chicago that had an eosinophilic increase in eosinophilia, and she subsequently came here and she founded the organization, the Campaign Urgent Research of Eosinophilic Diseases, called Curing, and today they've been supporting our work with over $5 million and certainly supporting other groups as well in the U.S. and have become a real driver and advocate for this disease. We subsequently worked, and today I'll just focus on the IL-13, which led to the basis for this drug, by Kareem Blanchard's work a number of years ago, showing that IL-13 overexpression in mice would lead to a type 2 response in the esophagus. And it was early work that we did, which was a very simple experiment that I thought it was almost like juvenile, but we took IL-13, we induced EOE, and then we gave an antibody against IL-13. And it was really not a very creative experiment, but the pharmaceutical people and thought it was very interesting, and it got published in what I call a weak journal, but it got referenced about 1,000 times in literature, showing that people took an interest in this particular finding, which was the ability of a humanized antibody, CAT354, to block the effects of human IL-13 in mice. We subsequently, in 2016, became fascinated with the possibilities of molecularly probing human allergic inflammation and the ability of genome-wide analysis to uncover pathogenesis. There was early work that we did with biopsies that we were getting from these patients. It was the first time we could probe human allergic disease at that level by doing genome-wide analysis. So we identified what we called the EOE transcriptome, which was work done by Kareem Blanchard, where she identified the signature of genes, which was several hundred genes. But one of the first things that was striking to us was we looked at the genes, and it was really like looking at under a microscope, but doing it in terms of the molecules. We saw coming up the IL-13 signature genes. And we postulated at that time that this transcriptome was induced by IL-13 overproduction, which she subsequently showed was taking place by showing that IL-13 was overproduced in the esophagus, and in vitro in cell lines we could recapitulate this with a cell from the esophagus, the epithelial cell, and induce the EOE transcriptome ex vivo with IL-13 alone. And that led to early discussions with pharmaceutical companies that I initiated on trying to convince them that IL-13 could be mediating this disease that most people had never actually heard of, and the companies weren't interested in. But there was this one company that was no longer interested in their anti-IL-13 that was sitting on the shelf. So there was a lot of controversy about the role of these cytokines that were initially identified in mice and their role in human disease. But we got hold of this antibody called QAX576. And this was a challenge actually to get a company to give you an antibody that wasn't FDA approved, when it wasn't being used, there wasn't a large amount of safety data, and also we had no funds. Cincinnati Children's provided the background, the environment, the great people, but they weren't necessarily funding the research, but we were able to leverage the philanthropy donations that were coming in from Cure and private donors to initiate a study with this antibody, which we gave intravenously in humans, adult patients here at the hospital, which was a single-site study that we initiated, and I held the IND for it. So that's a lot of work, and it took a couple of years. But you can see on this diagram, from the time we postulated the idea to the first infusion took 3 years, and then we carried out this study. Now, it was what I consider to be a very compromised study, because we didn't have the great people we have today. We didn't have the clinical trial office at the level it is today. We didn't have Dr. Kara Cleaver, who's the research manager. We didn't have the organization that we had today. But we did do this study. It was placebo-controlled, and we didn't have ways of really monitoring the patients, but I was very strongly convicted to the importance of the EOE transcriptome, because that, to me, was the signature genes, and it was completely objective, and we had ways of quantitating this. So we gave this drug to the patients, and we showed that the transcriptome was reversed. And the patients didn't jump up from joy. They didn't stop with their esophagitis to a large degree, but they did have a decrease in the eosinophils, and the transcriptome was improved dramatically, and that allowed us to map out in humans what I call the IL-13 nodes in humans, which was amazing because we were doing this in patients, which was just so cool for me rather than doing it in mice. And we postulated this, and we got it published in a pretty good journal, but we didn't hit the clinical endpoint, which is to make the patients feel better. And there were a number of reasons for that. It was certainly a small study, and it wasn't really our goal at the time. But that data in that paper was very convincing to groups, and people that understood this, not necessarily superficially, but Regeneron is a smart company, and they looked at this data, and they said this was of interest, and so did a couple other companies like Receptos in San Diego. And around that time, we also formed the Consortium of Eosinophilic GI Disease Research, which is ongoing today. It's an NIH-supported network that we lead here at Cincinnati Children's, and involves 14 other great centers in the U.S. that are working on these diseases. And we developed in Sager, along with other places, what we call clinical outcome metrics to basically monitor this disease, along with now Mauricio and his data management center here associated with the RDC, or the Rare Disease Clinical Research Network. We developed ways of clinically monitoring patients through validated metrics. And that was now positioning the companies to begin to organize around this disease, because they thought that the actual possibility of proving this theory would work. And at that time, there was a lot of advocacy going on. In addition to the Consortium of Eosinophilic GI Disease Researchers, we had partnership with patient groups besides Cure, such as the APVED group, the American Partners for Friends of Eosinophilic Diseases, as well as the Eosinophil Family Coalition, which was founded here at Cincinnati Children's by Melissa Scott. And these groups also pushed forward. So they formed a number of things, including bringing this to the attention of the NIH, as well as the federal government. And lo and behold, they were clever enough to get National Eosinophilic Awareness Week established in Congress, and it was written into legislation. So there's a lot that happened during that time. Now, we subsequently were continuing in our what I consider to be the more fundamental research, and I can say that we did the genetics of eosinophilic esophagitis through GWAS approaches. one of the top hits was a gene that we had never heard of before, which turned out to be an esophageal-specific intracellular protease, and Leah Cotton and Lisa Barton and others were heavily involved in this and did most of the hard work in terms of the genetics. But the main gene that we got out of that was the calpain 14, and then we, Lisa and Leah and others, showed that IL-13 was actually inducing this gene. So we connected the adaptive immune system, which is the IL-13, to the innate immune system, particularly in the esophagus, and we showed this connection. So this provided what I consider to be substantial data that this was a pathway that was going to be important. At that point, a number of companies had antibodies against IL-4 receptor alpha, and as well as IL-13, IL-4 receptor alpha being the IL-13 receptor, and they pushed forward in collaboration with us, but certainly people all over the world, to run large-scale clinical trials with the goal of getting drug approval. And that included the phase 2 trial with 2 different drugs, including the Dupilumab, and they were both positive, very nice positive results. And then in 2018, Regeneron pushed forward with their phase 3 trial, which involved a number of sites, across the world. In Cincinnati Children's, especially under the leadership of Sandy Durrani and Kara Cleaver and her group, we enrolled the largest number of patients at any site in the world, and that led to recent completion of that trial in 2020 with positive results, which is still ongoing, an extension of that study, and now a study that's in young kids, actually 1 and over, in addition to the original study, which was done in 12 and over individuals. So there's a lot that's been happening, and we were surprised on last Friday because we knew that the company Regeneron, which we're not directly connected with, of course we're research collaborators with them, but we have no stake in the game, had submitted a request for the drug approval. And this came in the setting of a recent number of drugs that were rejected for this disease because of various components really related to the data, and that was showing that the eosinophilic ablation in humans wasn't actually improving the patients as much as was expected. So there was a lot of downer about this disease. And the FDA was working closely with us, and as you probably know, we reported with them a number of papers on this disease in collaboration with the FDA. And then the FDA came out with guidelines for how controlled trials should be done, which we didn't necessarily agree with everything that they said, but it was some sensitive moments with the FDA. Nevertheless, this data that came out with the Dupilumab in the phase 3 trial was very, very positive, showing that it does improve patient symptoms, that it does improve endoscopic findings, and it made the histological features, which was using the scoring system that Ziegler and Margaret Collins had developed, was very dramatically improved compared to the placebo. And the EOE transcriptome, which they used exactly hours, they did it independently, was reversed by their drug, which proved that it worked. And then the FDA, interestingly, last week, at the end of National Eosinophilic Awareness Week, which was last week, Friday at 2:40 p.m., surprisingly issued a statement that they approved the drug, which I think they really did cooperate with us in general. It took 2 months for them to make the decision. They released it at the end of the National Eosinophilic Awareness Week, and it was right before the national meeting for the Digestive Disease Week, which was taking place in San Diego, so the timing was very nice. So we have this drug now, and it is indeed a victory for the patients. For the hospital, I think it's important that the hospital played a critical role. It's important for science because this came out from scientific investigations, and certainly the whole IL-4, IL-13 Th2 pathway. I want to acknowledge Nero, she stepped out. She's been working on this along with others here at the hospital. We did have a program project grant on IL-13 that we had for a number of years when Marshall Willis Park was here. So there was a lot of science that went into it, along with Fred Finkelman, that was doing a lot of work on this whole pathway, but we've really worked on the eosinophilic esophagitis component. So say that this is a victory for the hospital, for science, for advocacy, and for patients. And we think that this is the beginning of a series of drugs that hopefully advance patients. Here at Cincinnati Children's, we are not just interested in development of the drugs, getting it out there, but ultimately improving patients' lives to the highest level, and we anticipate that we'll need other drugs, and there a lot to be learned, which Bill Putnam always reminds me of. Very recently, you can see our website where he basically released statements about the conditions for potentially being considered for this drug, which will be costly, very costly. I don't know the amount, but it'll certainly be over $50,000 a year, which is a whole area of science and advocacy that others will work on. But there's a lot to do, and ultimately, like the Cure organization asked us to do, cure the disease rather than develop other treatments, which is an injectable medication that should be taken once a week. So I want to thank the colleagues that we've had over the years, because if I get credit for this, it's a circumstance, but there's so many people that are contributing. There's a huge amount of great people in this room, and people that have been around for a long time and have left, doing great things, contributed in huge ways. I want to acknowledge people at all levels, including every level. It starts off with administrative assistants that have just played a critical role in helping me and helping everyone carry this on. Amy Lawton is here, and there's been many before her as well who have had essential roles. There's been great structure here at the hospital. I can say that our IRB here is one, I think it's a great IRB. They're patient, they're friendly, they're cooperative, they understand things, they're allies, and this has been important. We are the single IRB central site for a lot of places in the U.S., and that's because we have a great infrastructure here for researchers, what the hospital invests in, including the cores here that have really made things that we've done successful, like the transgenic core, as well as the sequencing core and the single cell core. I want to thank especially my lab folks, and especially the long-standing lab manager, Julie Caldwell, who's, Dr. Caldwell got a PhD from across the street in molecular genetics and has been here, doing amazing stuff, understands things at a very high level and contributing in fundamental ways. I want to thank Julie Daisy for managing the Cincinnati Center for Esophageal Diseases for a number of years. Very complex interaction with a couple of dozen probably people now, and all the people who work in the CCAD. I want to certainly thank all the GI colleagues. This is a full partnership with the Division of GI, certainly pathology, but the other GI doctors, Vince Picata and recently Scott Bolton, and certainly the allergy people that have been working on this, including Dr. Asad, who's been working on this for a long time in parallel with us, as well as other people like Justin Schwartz and Eric Kellner, and certainly, as I already mentioned, Sandy Durrani and so many other people. I want to thank the funders for this work. Of course, NIH has been very critical for our work, and I want to thank the organizations, and some of them will speak now, but particularly acknowledging the Cure Foundation, the Bunning family, David and Denise Bunning, and Mike and Amy Ciccarello, have been very major supporters, but so many other people, whether it be just a pat on the shoulder, or encouraging a smile, or sending in a $10 check, and certainly so many other people. I want to thank the company Regeneron for pushing this forward, and there's a lot more to do. As you know, we're running another study that I hold the IND for, which is looking at Dupilumab eosinophilic gastritis, not eosinophilic esophagitis. And this is much harder to work with because there's about 1/10 the number of patients that we have here. There's a lot to figure out, and there's still need to develop better treatments, even though this is a major step forward. So there's a couple of people online. I think, who do you want to start with? Ellen? Let's do that. Ellen Kodroff? What's the best way to get her going?
Hi. You want me just to start?
1 second, you just want to make sure your video's on.
Okay.
You know the best way to get this off?
Stop sharing.
Share.
Okay.
Okay, Ellen, it's all yours.
Hi, thank you so much for just inviting me to this very exciting day and giving me the opportunity to speak. As Dr. Rothenberg said, I brought my daughter there in 2003, and never really had dealt with any rare disease or research, and just thought hospitals funded research. I was pretty naive to the way things work. And when Dr. Rothenberg and Dr. Putnam explained there was no cure and there really wasn't any good treatments, I very distinctly remember looking at Dr. Rothenberg and saying, how much do you need to start your research or to further your research? And at the time he told me $1 million would be a really good place to start. And I looked at my daughter and looked back at him and said, somehow, I don't know how, but we will get you the $1 million. And since then, we haven't stopped. We've now donated over $6 million to research, but our faith is in the Rothenberg Cured Lab. We're so honored to have our name shared with your lab there at Cincinnati and your work. We are just dedicated to continuing to fund the research with your lab. We are so impressed by all the work that is done there. Your dedication is like no other. We just know and believe that one day there is going to be a cure and a better treatment because of the work you're doing. And we're very honored to be a part of this research and to be a part of your lab. I can remember our first lab tour of your first lab, and I think there were 3 or 4 people in there. So to watch it grow to where it is now and to see the work coming out of this lab is just rewarding as a patient voice and a patient advocate, and for not only for my daughter, but I just know the excitement of every single patient dealing with this disease, and I saw it by our Facebook and how much it increased this week by sharing all this exciting information. We reached 143,200 people this week on Facebook about this disease and about the work that you've done and other researchers and Seeger. So we're just so honored and so proud to be a part of it. And we hope that you know that you'll continue to get our support, and we'll see many more great things come out of your work at Cincinnati Children's Hospital. So thank you for letting us be a part of it.
Thank you. I'd like to have Amy Zuccarello from the Assemble Family Coalition say a few words. Amy?
Hi, thank you so much for having me here today. First of all, I just want to share a few words from Melissa and Kathy, the co-founders of Eosinophilic Family Coalition, and then I have a few personal words I'd like to share. Since the very beginning, the CCD has been a trailblazer in Dr. Rothenberg in both research and treatment of eosinophilic disorders, and is an inspiring to think about the work done at the center that has had a profound impact on people around the world. All of the families that the EFC has interacted with over the years, and how we have seen even patients who have never been treated at Cincinnati have truly benefited from the work done here. And the CCD has been a constant beacon of hope that we knew would reach this very important day. Beyond medical treatment, the center has always thought holistically about the patients they serve. The collaboration with the EFC to create Camp Courageous is a perfect example of this. The CCD staff have always considered the patients they treat for more than the sum of their symptoms, and for that we are grateful. We're grateful for Lab Day. We're grateful for all the interactions with the patients to help them understand how important clinical trials are, how important research is. Without patients and without Dr. Rothenberg and the team and all the doctors listening to the patient voice, without that collaboration between the two, the clinical trials wouldn't exist and this day would not have happened. So we're forever grateful for that. Personally, I just want to say that my family is very honored to be able to privately donate to Dr. Rothenberg in the lab. Dr. Rothenberg, you're a leader, you're a friend, you're a doctor that is absolutely, the whole team at the CCD is, where our family is fully amazed by you, that you wake up every day, you dedicate your life to changing the outcome for our children. And it means so much to us that this isn't just your job, it's your passion. My son Luke is now 16 years old. He was diagnosed at Cincinnati Children's at 18 months. We live in Dallas, Texas, and I remember very vividly the gastroenterologist at Children's in Dallas looking at my son and saying, "We've never seen this before, but we're hearing of a group at Cincinnati Children's that's looking at eosinophils, and we think you should go there." And that's where we went, and that's where we continue to go. And he's 16 now with 15 safe foods. He's tube fed, and we are hopeful that this FDA approval will be a life changer for him and so many other kids. So thank you, Dr. Rothenberg, for your leadership, for everything you do, and our family will continue to support you. Thank you.
Best wishes to your family and your community there in Texas, Amy. MJ Strobel, Mary Jo Strobel from the AppFed organization, would you like to speak?
Hi, yes. Thank you for the kind invitation to join and have the floor for a few moments. It was indeed an exciting week last week. The news of Dupilumab receiving FDA approval for EOE was such welcome news, and to have that information come across during National Eosinophil Awareness Week just made it that much sweeter, didn't it? 15 years ago, AppFed set out to establish an official awareness week, which was formally recognized by the U.S. House of Representatives. The purpose of the week was to really double down on efforts to increase awareness and educate patients, the general public, and the medical community about eosinophil-associated diseases. With greater awareness, the need for research is amplified, and a supportive community develops. So I think this year's Awareness Week is going to be pretty tough to top in the future. When AppFed launched its research program 5 years after our founding, it was to support projects that advance research in eosinophilic disorders through a rigorous peer-reviewed process, and many of the grants we awarded involved research of the immune and inflammatory pathways that played a role in the development of EOE to help guide potential future therapies. We're grateful for the donations that families have contributed to our collective community that have accelerated advances in the field. 20 years ago, there was so little known about these conditions and little research being conducted compared to today. Today, we can say we have an indicated therapy, which feels like lightning speed, and we've each played a central role to get us there. By working together, our collaborations have driven transformative change, and this recent FDA approval is the outcome of years of hard work. From the critical research work spearheaded and conducted by Cincinnati Children's, other institutions throughout the U.S., the critical involvement of patient groups that have fueled the research dollars, advanced federal funding, medical codes, and have engaged the community to support research efforts has just been exemplary. While we all deserve to be very proud of these achievements, we are proudest of the real heroes, which are the patients and families who have selflessly and bravely participated in research and clinical trials so that development such as Dupilumab could advance. So we hope that this is one of the first of many therapies that will be discovered as a result of ongoing research in the field of eosinophilic disorders, and we look forward to the next chapter in the EOS story. So thank you.
So I just want to close by thanking some further people, especially the people who put this event together today. Mel Engler, who's behind so many things in the division and the success that we have. Carol, Heather, and others. And I want to also put in a thank you to Shauna Heidinger thank Kevin Titus for being a partner in the division. Truly, it's co-leadership, and Kevin. Bill, see right? Dr. Butler. See in the room? No, please. Say a couple words.
Well, thanks everybody for coming to this event, which we're all very proud of. It's a culmination of 2 decades of work toward managing eosinophilic esophagitis from the ground up, I suppose you should say. I think we're all indebted to Mark and his incredible team for all the basic science research that's gone on here. And but I also want to recognize that it's been a clinical program supported by the hospital and the institution and the division, patients are taken care of by a collaborative group with the allergists and with the dedication of our nurses and dietitians and psychologists. We're able to provide clinical care in addition to the research side of things, which has been crucial. I want to echo what Mary Jo said, that we're totally indebted to our patients who come here for clinical care, but then decide to participate in research, which makes all of the clinical trials relevant and, in this case, successful. So, thanks everybody for coming, and see what the next decade brings.
Billy, in the beginning, when I thanked you and the partnership that we've had since 1999, when you were the person who approached me and your excellent team and all the other GI doctors. It's critical. I just wanted to say, but one more thing which to emphasize is that this study, this phase 2 and phase 3 study, many of you walk by the door and you see Dr. Collins in there, Margaret, and she's there on weekends, and it's amazing. She counted every slide in the study. She was a single pathologist for all the Regeneron studies. Every slide went through her microscope. She sat there and counted the eosinophils on every single one of them. And one of the things I hope it doesn't happen, Kara, but we did not get an FDA audit, and they actually approved the drug. And now it's a single-site pathology center. Hopefully we won't see the FDA here. And Mal can speak about that because she's seen the FDA before, but we haven't seen them yet. But they did approve the drug, but it doesn't mean they still won't visit. But the point is, Dr. Collins did every one of those slides that was used for the approval. Thanks for gathering today. Let's keep it going strong. There's a lot to continue to do.
We have to go on.